everybody. Today we're going to be talking about the steps in cell division. And so if you do have your study guides, you can bring those out, and also your clickers, and you can follow along as I go through the lecture. So why do cells divide, and why do we care? Well, we start out life as a single cell with a copy of chromosomes, complete copy from our mom and our dad as a single cell. But as we go through life and through development, from infancy to adolescence to then adulthood, we actually wind up with an average of about 100 trillion cells. So through those stages, we've obviously gone through a significant amount of cell divisions. So cells dividing, it's important, and we care about it. So any thoughts from you guys on some functions of cell division? Why is cell division important in an individual's lifetime? Yes?
Repairing the damaged cells.
Definitely repair of tissue damage. Other thoughts? Why cell division is important? Yeah?
Passing on genes to the next generation.
So if we want a faithful passing of genes, let's say we're growing tissues, so in growth of tissues, we want one skin cell to have the exact same copy of the genetic material as the other. These are the real important functions. Any others that come to mind? Yeah?
Genetic variation.
Genetic variation, probably more the case in meiosis, and that's something we'll get to later today, but in terms of cell division, any other thoughts? Yeah, that's pretty much what I was thinking, so you guys said it. Growth, an example is, let's say you want your long bone of your leg, your tibia, to get longer. That's a result of cell division in your bone cells. Development, as we go through, let's say, reproductive maturity, we need to undergo mitosis to get cells that will do that process. And as you said earlier, tissue repair, if you get a cut in your skin and you need to replace those skin cells, cells are very active in their division. So all of these are reasons why we go through cell division. And so let me talk to you a little bit about the stages involved in cell division. And we're talking about a eukaryotic cell. The cell it divides in a cycle, and if we're talking about a typical skin cell, that cycle takes approximately 24 hours. And I will say that 23 of those hours are spent in interphase, which is a phase of the cell cycle where the cell is basically preparing to divide. Not actual division yet, but a preparation of division. So 23 of those hours is in interphase, getting the stage set for cell division, and only 1 hour of that is the actual division itself. And so let's talk about those different stages. So in interphase, it starts out with G1 or gap 1 phase, and this is usually the longest stage of the cell cycle and where the primary growth is taking place. If it's a liver cell, this is the time when that liver cell is detoxifying drugs and antibiotics, for example. This is where the cell is going to spend the majority of its time. So I'll ask you to get your clickers, and on your sheets, if you could draw a cell that's in G1, somatic cell being one that's not involved in reproduction. It's got a diploid number of 4. How many maternal chromosomes are there? If you could register your answers now. Okay, I think we've got a good cross-section. So 74% of you said 2, and that is the correct answer. So in a somatic cell, with 1 copy from each parent of a chromosome, 2 of those chromosomes are going to be from the maternal origin. Another clicker question for you. In that same cell, how many total chromosomes are there? And 87% got it right. There are 4 total chromosomes in that cell. So this is in G1. Let's progress throughout more of the cell cycle. We then go from G1 to S phase, and this is the replication of DNA. If growth signals tell that cell it's time to divide, it will commit to that division by going into S phase and replicating the DNA. Replication of DNA involves our homologous chromosomes doing their thing and copying themselves to the best of their ability without any mistakes. Sometimes mistakes happen, but we're trying to avoid that. As a result of replication, we get 2 copies of each chromosome that are now called sister chromatids. They're connected with each other via a centromere, and now we've got homologous chromosomes, meaning one copy from mom, one copy from dad, that have an exact duplication or two sister chromatids held together. So in that light, I'll give you another question. After this replication, how many maternal chromosomes are there now? To weigh in on that. All right, so this was a bit of a trick question. So after replication, there still are two chromosomes. Those chromosomes, however, are replicated. So those two chromosomes, that hasn't changed. It's just that now we've got a duplication of those chromosomes in terms of sister chromatids. Then in that same cell, how many total chromosomes are there? All right, so in that same cell, there are 4 total chromosomes, and again, those chromosomes are duplicated, so they each consist of 2 sister chromatids. However, that doesn't change the chromosome number, so we're not increasing the chromosome number. That still is 4. It's just that those 4 chromosomes consist of 2 sister chromatids after replication. Any questions on that? That all make sense? All right. Let's proceed through the cell cycle then into now the DNA is replicated, hopefully not any mistakes in that process. We proceed then to G2, gap 2 phase. This is where the organelles replicate. Remember that we are trying to prepare the cell to divide. We've got to have organelles in each cell, right? So the mitochondria, they'll divide by binary fission. We have the microtubules organizing and getting themselves ready to form the spindle apparatus. And we also have our endoplasmic reticulum and various other parts of the endomembrane system dispersing so that now our 2 cells can ultimately have an exact copy of all of the organelles as well. Once that's accomplished, 23 hours later, we can then proceed to the actual division, which is mitosis. Mitosis, and you'll see on your sheets that there's a place for you to either draw each of these stages or just write out the main hallmarks of each of those stages so you can look at those all together and see the comparison of the various stages. So prophase is first, and now that all the DNA has replicated, the DNA can now condense into these chromosomes that we typically see, that X-shaped structure of a chromosome. Those are now visual in the light microscope. In the lab, this past week, you may have seen these chromosomes visually as those X-shaped structures. They condense because that makes them easier to separate in the end. So that's a hallmark of prophase. Another is that the spindle apparatus, which has microtubules, that forms, that becomes distinguished. And the nuclear envelope has to dissipate. So in a typical cell, the DNA is housed within the nuclear envelope. It keeps it secure and safe. However, during the process of cell division, you want that nuclear envelope to dissolve so the chromosomes have access to the cell and can move to opposite poles as they get to that stage. The next stage is going to be metaphase, and this one probably you saw in the microscope most clearly. It probably didn't exactly look like this cartoon. It was a little bit more messy, but you might have been able to distinguish this stage pretty well. And this is a stage where chromosomes will align in the center or the metaphase plate of the cell, and they'll do so end to end. Another thing you'll notice that you probably weren't able to see in the microscope, but you'll notice that each of these sister chromatids that have gone through the replication in the previous S phase, they're each attached to spindle fibers that orient them to opposite poles of the cell so that you can see what's going to happen next. Then they're going to be able to pull apart. But at this point in metaphase, they've just aligned in the center of the cell. Then we proceed to anaphase, and this is where the spindle fibers that are attached to those each sister chromatids are going to shorten like a fishing line that you're reeling in your fish. Those microtubules will shorten, and that will apply pressure to those sister chromatids, pulling them apart. Additionally, the poles themselves, which you can't really see in this image, but the poles themselves are moving further apart as well. So by the action of the poles moving apart, as well as the microtubules shortening, all of that is having the effect of pulling sister chromatids apart from each other. So in the microscope, you probably also saw sister chromatids that are starting to move to opposite ends of the cell. That's a hallmark of anaphase. Finally, we reach telophase, and at this point, the work has been done for the most part. The spindle apparatus has done its job, so it can just disassemble, it's not needed anymore. The nuclear envelope will start to reform around these chromosomes that have now separated. And you'll start to see cytokinesis, which is the physical separation. If that's an animal cell, that's happening via a cleavage furrow that's going to ultimately separate these two cells apart from each other. End result is that these cells can now undergo another round of the cell cycle as needed. So one final question and summary for you. What is the end result of one cell cycle? And far and above, you guys got it. It is that there are two cells with the same amount of genetic information. Hopefully they're genetically identical as long as there are no unrepaired mutations. And we can now have these cells as daughter cells to go through another cell cycle again. And so this is the visualization of that final result where we see two now independent daughter cells that are right at the beginning again. Once they get the signals to divide, they can go through and go through another round of division there. All right? So that's all for today. Any questions or points that I can clarify for you before we end? Yes?
Could you just draw what the number 1 would look like, the somatic stem cell before division?
Yeah, so going back to that first question I asked you. Right, so drawing a somatic cell. Before replication, so a couple things to think about here. The replication has not yet happened. So we don't see two sister chromatids. So we've got our somatic cell. I mentioned that it has a diploid number of 4. So all we would see there would be 4 unreplicated chromosomes. And so then they go through S phase. Once they do go through S phase, they'll look like this. But either way, they're just called chromosomes before and after replication.